Okay, students, so this is Industrial Biochemistry, and the course code is BCHM-351. This is lecture 5, and the topic is acetic acid production by fermentation. I am Dr. Sadiq Ibrahim, Assistant Professor, Department of Chemistry, Cohort Ministry of Science and Technology. And you can access these lectures by logging into your account of KCMS, and from there you can find the relevant link for the West Media Server portal, whose website is ms.cohort.edu.pk. Now the topics to be discussed, so they include acetic acid bacteria, anaerobic process by which the acetic acid is produced, the industrial production of acetic acid, and different methods, biological methods which are produced for the production of acetic acid at industrial level, and uses of acetic acid. Now moving towards the acetic acid properties. So the acetic acid has a molecular weight of 60.05, and it is a principal constituent of vinegar. So you all know that the vinegar has around 4 to 8% of acetic acid present in it, and the rest is water. The first vinegar was spoiled wine. Now, consisting the Latin word acetum means sour or sharp wine. So basically, acetic is from a Latin word acetum, which means sour or sour. It is also known as ethanoic acid, ethylic acid, vinegar acid, and methane carboxylic acid. The glacial acetic acid is a pure compound which has a purity of up to 99.8%, as distinguished from the usual water solutions known as the acetic acid. So glacial acetic acid is the purest form of acetic acid available for experimentation purposes. The boiling point of acetic acid is 118 centigrade, and the melting point of rhombic crystals is 16.6 centigrade. The glacial acetic acid is highly corrosive to the metals, so it means that it can cause damage to the metals. Acetic acid is soluble in alcohol, miscible with water, glycerol, ether, acetone, benzene, carbon tetrachloride, and it is insoluble in carbon disulfide. Now moving towards the structure of the acetic acid. So its molecular formula is written like this: CH3, C double bond O, OH, and if you write it in a line diagram, so this is the methyl group which is connected to the carboxylic carbon. The carboxylic carbon is then connected to the carbonyl oxygen along with it to a hydroxyl group. So as there is a methyl group attached, so that's why sometimes the acetic acid is referred to as a methane carboxylic acid. Now the acetic acid production. So it can be produced in the factories by using three major methods, and they are the chemical reaction, in which the liquid and vapor phase oxidation of petroleum gases is taking place along with the catalyst, as well as the oxidation of acetaldehyde. It also produces acetic acid. Production from fossil fuels is the second method, in which the acetaldehyde oxidation gives you the acetic acid as a final product. The hydrocarbon oxidation also gives you the acetic acid, along with the methanol carboxylation, which leads to the production of acetic acid. So the topic of our interest is biological processes, and basically the acetic acid is produced by the process of fermentation. So there are two types of fermentation. One is aerobic and the other one is anaerobic. So in aerobic you have presence of oxygen available to the fermentation process, and in anaerobic you don't have the presence of oxygen or provision of oxygen in the process. Now moving towards the acetic acid bacteria. So there are gram-negative bacteria belonging to the family Acetobacteriaceae, as well as to the alpha subclass of Proteobacteria. And the recognized genera are Acetobacter, Saia, Echidomonas, Glucanobacter, Glucanotobacter, and Kozikia. With the exception of Saia, these bacteria, they produced large quantities of acetic acid from ethanol and can grow in the presence of 0.35% acetic acid as the starting inoculum. Now moving towards the anaerobic process by which the acetic acid is produced. So it is produced by a two-step process. The first step is the production of ethanol from a carbohydrate source, which is normally glucose. The optimum temperature is maintained around 30 to 32 centigrade, along with the usage of anaerobic yeast, which in this case is Saccharomyces cerevisiae. Now in the equation you can see that you have a molecule of glucose, which is broken down into two molecules of carbon dioxide and two molecules of ethanol. Now in the second step, oxidation of ethanol to acetic acid is taking place, and a variety of bacteria can produce acetic acid. Now the only members of Acetobacter which are used commercially is Acetobacter aceti, and at a temperature of 27 to 37 centigrade. So basically at commercial level, Acetobacter aceti in the form of bacteria is the most common and preferred source of bacteria used for the production of acetic acid. Now in case of anaerobic process, the theoretical yield is 0.67 gram acetic acid obtained from 1 gram of glucose, and here the glucose used is 100% pure. Now the realistic yield is of 76%, that is 0.51 gram acetic acid is produced from 1 gram of glucose. Now the anaerobic process for the production of acetic acid requires 2 pounds of sugar or 0.9 pounds of ethyl alcohol or ethanol per pound of acetic acid to be produced. Now, the complete aeration and strict control of the oxygen concentration during fermentation process are important to maximize yields and keep the bacteria viable. Now, in relation to the anaerobic process or the production of acetic acid, in the 1980s, this process emerged based on the anaerobic fermentation using the Clostridia genus of bacteria. Now, the commonly used bacteria, they are Clostridium aceticum, Clostridium thermoaceticum, Clostridium farmacoaceticum, and Acetobacterium woodi. Now, it is an obligate anaerobe, gram-positive, spore-forming, rod-shaped, thermophilic organism with an optimum growth temperature of 55 to 60 centigrade, and optimum pH is around 6.6 to 6.8. Now, the Clostridia genus of bacteria can convert the glucose, xylose, and some other hexoses and pentoses, along with examples of fructose, lactate, formate, and pyruvate, almost quantitatively into acetate according to the following reactions. Now, here you can see that you have a hexose, and which is being converted into 3 molecules of acetic acid. Now, the Clostridium thermoaceticum is also able to utilize 5 carbon sugars like the ribose sugars, and here you can see that 2 molecules of pentose are converting and producing 5 molecules of acetic acid. Some acidogenic bacteria reduce the carbon dioxide and 1 carbon into acetate as well. The anaerobic route should have a lower fermentation cost than the aerobic process. So that is very important. Theoretical yields, like based on the calculations on the paper, 3 moles of acetic acid is produced from 1 mole of glucose consumed. That is 1 gram of acetic acid is produced per gram of glucose. The overall reaction can be written as follows. You have a hexose molecule plus 2 water molecules, giving you 2 acetic acid molecules plus 2 carbon dioxide molecules and 8 protons. And then the 2 acetic acid molecules along with the 2 carbon dioxide molecules and the 8 protons give you acetic acid plus 2 water molecules. Now, the enzymes involved in the production of acetic acid. So, tetrahydrofolate enzymes, carbon monoxide dihydrogenase, which is abbreviated as CODH, NADP dependent formate dehydrogenase, for example FDH, and cholinide enzyme. So these enzymes are involved during the generation of acetic acid. Now, these enzymes are metalloproteinic in nature. For example, the carbon monoxide dehydrogenase contains nickel, iron, and sulfur in its structure. FDH contains iron, selenium, tungsten, and a small quantity of molybdenum as well. And the cholinide enzyme, which is a kind of vitamin B12 compound, it contains cobalt in its structure. Now, in most typical batch fermentations processes, cell concentration initially increases exponentially and then decreases. This we have discussed in detail in the process of fermentation lecture. Towards the end of the fermentation. Acetate concentration also increases and then levels off, which means that it becomes stagnant. High glucose concentrations inhibit the initial growth of Clostridium thermoaceticum. However, after adaptation, the fermentation proceeds rapidly. Now, there appears to be a minimum ratio of nutrient concentration to glucose concentration to produce acetic acid. Now, in anaerobic process, if glucose is still available but the nutrients is not, the microorganism will produce byproducts such as fructose. Acetate production from glucose by Clostridium thermoaceticum generates 5 mole of ATP per mole of glucose consumed. Now this results in high levels of cell mass per mole of glucose consumed. To maintain the productivity, the cells must balance their ATP supply and demand. So there should be a balance between the supply and demand of the ATP. Since growth of Clostridium thermoaceticum consumes more ATPs than maintenance, most of the acetic acid produced during the growth phase. Now, when cell use yeast extract as a source of amino acids, the nucleotides and fatty acids, they will need less ATP in comparison to if they have been synthesized these compounds using ammonium ions as the starting material. So it means that the yeast extract is a better source of nitrogenous compounds in comparison to the ammonium ions directly being used. Now, in case the assimilation of ammonium ions is important, if the cells are able to recycle the ATP generated during the production of acetic acid. So the regulation of ATP is important during the production of acetic acid. The ammonium sulfate, another source of ammonium ions, is a cheaper nutrient and it could partially replace yeast extract without resulting in formation of byproducts such as fructose. Medium cost could be lowered further by substituting corn steep liquor for yeast extract. So as the corn steep liquor, it is more cheaper in comparison to the yeast extract. So then by using the corn steep liquor, you can decrease the cost of the process. Now the biological pathway for the biosynthesis of acetic acid, and in this flow sheet diagram you can see that in the start you have the glucose as the starting material and it is being converted into lactate, ethanol and pyruvate to different pathways. So here we are more interested in the acetic acid, so we will focus on this pathway because the rest of the pathways they give you propionate, acetate, carbon dioxide. So when the glucose, as a result of metabolism of glucose, you are getting pyruvate and the pyruvate is converted into ethanol and then from ethanol by the oxidation process it is converted into acetic acid. So this is the biological pathway by which the glucose, which is the starting material, and acetic acid, which is the ending material, is obtained as a product in the biological pathways. Now the industrial production of acetic acid. So there are many methods which are used for the production of acetic acid in different types of industries. So first one is the let alone method. The second method is surface fermentation. The third one is submerged fermentation. And the fourth one is the batch fermentation. Now the first method, which is the let alone method, the industrial fermentation process have evolved from the simple let alone method, which involves a partially filled open container of wine exposed to air to the field, and fermentation in which a series of casks are filled with wine and inoculated in series by the vinegar produced in the previous casks. So it is a type of cascade process. Now the second process or method by which the acetic acid is produced in the industries is the surface fermentation method, and it is basically known as the trickling or German process, in which the microbial population is attached to an appropriate support, usually the beechwood shavings, and the product is trickled down while a large volume of air is sparged up through the bottom of the tank. Now, this process was the basis for the manufacturing of a trickling generator that incorporates forced aeration and temperature control. The partially converted solution collects at the bottom and is cooled, pumped back up to the top, and allowed to trickle down until the reaction is completed. So it is a kind of slow process, but repeated batches can get you the maximum yield. Now, in terms of the surface fermentation, there are some disadvantages and advantages for this method used. So the advantages, they include the low cost, ease of control of the process, high acetic acid concentrations means high yield, and lower space requirements. The disadvantages, the cost of the wood shavings, that is very expensive, long startup time, takes a lot of time, loss of ethanol by volatilization, and production of slime-like material by the Acetobacter, for example the Acetobacter xylem, are some of the disadvantages of surface fermentation process for the production of acetic acid. Now the third one is the submerged fermentation process by which the acetic acid is produced. So in 1949, Baumgartner and Ebner applied submerged fermentation technique for the oxidation of ethanol to acetic acid. Now the level of gas phase, the oxygen is crucial to this process, and thus efficiency is based on broth aeration with oxygen. So it is a kind of aerobic process. For industrial purposes, 10 to 18% ethanol, and 5 times the nutrients are used for the submerged fermentation, and these are the starting conditions for the fermentation process. Now, when the concentration of ethanol reaches from 0.4 to 2.5 gram per liter, 50 to 60% of the solution is removed and replaced with fresh substrate containing 10 to 18% ethanol. And as a result of this process, nearly 80 milligram of dry bacterial solids are produced from 1 liter of the culture medium. Now, in case of the submerged fermentation process for the production of acetic acid, the theoretical yield is 1.7 to 2.1 grams of acetic acid produced per liter per hour in a semi-continuous batch. And during the production of the acetic acid, the dead cells of the bacteria, they cause foaming. So in order to remove this foaming, mechanical defoaming techniques are used. Now, compared to the surface fermentation process, the submerged fermentation results in higher productivity, faster oxidation of ethanol, smaller reaction volumes, low personnel costs due to automation of the process, and fewer interruptions due to the clogging by shavings, and lower capital investment per product amount. So these are the advantages of submerged fermentation method in comparison to the surface fermentation method, which doesn't have these advantages. Now move towards the batch fermentation process for the production of acetic acid. Now a draw and fill bioreactor in combination with the membrane appears to be the optimum design of the batch fermentation tank. Now at the end of the fermentation, a portion of the fermentation broth is withdrawn through the membrane module. The cells are recycled and the reaction vessel is charged with fresh substrates. In batch fermentation, without self-recycle, acetic acid productions is proportional to the amount of yeast extract and trace salts supplied in the medium. Now, for all types of bioreactors studied, increasing dilution rate increases the volumetric productivity but decreases the specific productivity, that is the grams of acetate produced per gram of the cells. Now moving towards the downstream processing and cell separation, which we have discussed in the earlier lectures in detail, that what is downstream processing. So to isolate, to purify and concentrate the product often determines the economic feasibility of the process. Now the first operation is cell separation and cell lysis, which can be done by cross flow, microfiltration, nanofiltration and electrodialysis, and these are useful in concentrating the glacial acetic acid. Solvent extraction with distillation is the preferred method for chemically derived acetic acid, whereas freeze concentration is used for vinegar. Furthermore, if the acetate is required in the free acid form, there will be additional cost to convert the salt form produced in the anaerobic fermentation into the free acid form. There is a liquid-liquid extraction step which is used to recover acetic acid from the chemical manufacturers of cellulose acetate, vinyl acetate and other acetate products. Now the extraction efficiency is high when the organic acid is present in the undissociated, that is the acid form, specifically at a low pH. Because at low pH, the acetic acid will get protonated and it will be in the undissociated form. Now moving towards the uses of acetic acid. So first of all, acetic acid is being modified into different types of chemical compounds. For example, vinyl acetate, which is used in textile finishes, vinyl plastics, latex paints, and adhesives. Another modified form of acetic acid is the chloroacetic acid. Then you have the acetic anhydride, which is used in pharmaceuticals and plasticizers, as well as the acetic anhydride is further modified into cellulose acetate, which is then used in molding compounds, transparent sheets for textile fibers, in the synthesis of photofilm and lacquers. The acetate esters is also one of the derived form of acetic acid, which is used in as a solvents. And directly the acetic acid is used in textiles, pharmaceuticals, and mainly for the preparation of synthetic vinegar. Now the acetate salts, which are formed by the reaction of acetic acid with acid and base, so they give you acetate salts, which are used in heat transfer liquids, used as a meat preservative, also used as a neutralizer, fungicide, and for the purpose of de-icing. Now the references which need to be followed for further understanding and for further reading are mentioned here, and the serial number 5, the book with the title Principles of Fermentation Technology, is very relevant to this topic, and it is written by Peter F. Stanbury, Alan Whitaker, and Stephen Hall. So if you want to further grasp the idea of continuous and batch type culture techniques in fermentation, so this book is very relevant for understanding and clearing your concept. Thank you.